so recap of glycolysis. Glycolysis is anaerobic. It does not require oxygen. It happens in the cytoplasm of the cell. The point of glycolysis is to, if oxygen is present, is to get these pyruvic acids and shuttle them into the mitochondria for aerobic cellular respiration, the citric acid cycle, and the electron transport chain. 3 phases. Our energy investment phase. This is where we utilize 2 ATP. We put in energy. Then we have our cleavage phase, where we take our 6-carbon glucose and we cleave it into 2 3-carbon molecules. Each one of these will enter into phase 3, which is the energy harvesting phase. And with both of these 3-carbon molecules entering into this phase, we get 4 ATP back, which gives us a net gain of 2 ATP total. We also get 2 NADHs, which can't be used for energy right away, but they are high energy electron carriers, and they can be shuttled into the mitochondria and enter into the electron transport chain, and we can get energy that way. And we get our pyruvic acids, which can go into the citric acid cycle. If oxygen's present, pyruvic acid goes to the mitochondria, enters the citric acid cycle. If oxygen is not present, well, 2 ATP is better than no ATP, so if oxygen is not present, we enter the lactic acid cycle. We convert pyruvic acid into lactic acid, and we convert that NADH that we just created, we transform it back into the NAD+, meaning this can go back up to these steps, and we can make more ATP. And so it just continues. That lactic acid cycle, very important in muscle metabolism, and so this will come back up, okay? So the point of lactic acid formation is to regenerate these NAD pluses, which can go back to the energy harvesting phase, and we can get more ATP, okay? So oxygen is present, so let's talk about what happens when that pyruvic acid enters into the mitochondria. You'll notice that in these PowerPoint slides, I do have a lot of text, and this is for if you want to read on your own. I'm not going to read all of the text that's on the PowerPoint slides. It's essentially what's in your textbook. I'm just going to go to the big pictures and show you and describe what's happening. So let's move on to the citric acid cycle. So what this is showing you, this is very not to scale, but this outer light orange square, this would be a cell, and where pyruvic acid from glycolysis is, that would be in the cytosol or the cytoplasm, okay? This inner darker orange square, this is the mitochondria, and you'll see that there are 2 areas to the mitochondria. A mitochondrion, singular, mitochondria, plural. A mitochondrion has 2 membranes, this outer membrane and this inner membrane. Between the 2 membranes, this orange area, this is called the intermembrane space. Okay? You'll notice that the inner membrane is very, very highly invaginated. The reason for that is because the proteins that carry out the electron transport chain are embedded in this inner membrane. So this folding gives us more surface area, meaning more electron transport chain machinery, okay? So the more inner membrane we have, the more electron transport machinery we can have, okay? Now, this inner portion, this yellow portion of the mitochondria, that's called the matrix, and it's the inner matrix of the mitochondrion, okay? This inner matrix is where the citric acid cycle takes place, okay? So in order to start the citric acid cycle, we have to take that pyruvic acid from glycolysis and shuttle it into the inner matrix of the mitochondria. We have to convert the pyruvic acid into acetyl-CoA, okay? Into acetyl-CoA. And this transformation, okay, going from pyruvic acid to acetyl-CoA, this step gives us, we split off a CO2 molecule, and if you look, remember the pyruvic acid is a 3-carbon molecule, right? It's half of the glucose that we started with. One of those carbons gets split off as CO2. Remember, this is a waste byproduct that we will eventually have to exhale. The conversion from pyruvic acid to acetyl-CoA, we also generate another NADH. Remember, these are high energy electron carriers, can't be used for energy right away, but we can put them into the electron transport chain and make ATP energy that way. All right, so one pyruvic acid. So this is showing you what happens with just one pyruvic acid. But how many pyruvic acids did we get from glycolysis? We got 2. So from one glucose molecule, we would get 2 pyruvic acids, and all of these steps would happen twice, okay? 1 for each pyruvic acid molecule. Okay, so we have to convert pyruvic acid to acetyl-CoA, and in the process, we split off a CO2 and an NADH, which can be used in the electron transport chain. Now this acetyl-CoA, which is a 2-carbon molecule, can join with, now I'm not going to ask you to know oxaloacetate, okay, but oxaloacetate, okay, or oxaloacetic acid, either one of those names. You'll notice that oxaloacetic acid is a 4-carbon molecule, 1, 2, 3, 4. When it merges with that acetyl-CoA that we just created, we get our 6-carbon citric acid, 1, 2, 3, 4, 5, 6. 2 of those carbons being from the glucose, that beginning glucose, okay? So that's why this is called the citric acid cycle, is because the very first product of the citric acid cycle is a molecule called citric acid, okay? So this will then go through a series of steps within this cycle. And I want to point out, big picture, what are we getting from this citric acid cycle? You'll notice that 1, 2, 3 NADHs are formed from one pyruvic acid, okay? But we also got this NADH right here, okay? So going from pyruvic acid all the way through the citric acid cycle, 1, 2, 3, 4 NADHs, okay? All right, so 4 NADHs. Let's see, what else do we, oh, right here, look, we get 1 ATP, right? So this ATP can be utilized for energy, for reactions right away, but we only get 1 per turn of the citric acid cycle, so only 1 pyruvic acid. We get 1 ATP. We also will get this molecule right here called FADH2. FADH2 is also a high energy electron carrier, just like NADH, okay? We can't use it for energy right away, but it can enter into the electron transport chain and give us, and help with synthesis of ATP energy that way, okay? So let's go through if we were to, also, let's see. You'll notice that a carbon dioxide gets split off here, and another carbon dioxide gets splits off here. This eliminates all of the carbons from the original glucose molecule. So we completely oxidize glucose, right? Because look, here's our pyruvic acid, which has 3 carbons. We split off 1 here through carbon dioxide. We split off a second one here, generating another carbon dioxide, and we split off the 3rd one here, generating another carbon dioxide. So we use up all of the carbons from the glucose that we originally started with in glycolysis, okay? So let's see what we were putting in and what do we get out with the citric acid cycle. So from the conversion of pyruvic acid to acetyl-CoA, right? Remember, we have 2 pyruvic acids, so this would happen twice. From this conversion, we would get 2 NADHs, 1 per pyruvic acid, 2 NADHs. We would get 2 carbon dioxides, 1 per pyruvic acid, and we would get 2 acetyl-CoA molecules, 1 per pyruvic acid, okay? Now, each of those acetyl-CoA molecules, which are generated from both of the pyruvic acid molecules, would then enter the citric acid cycle, okay? And it's a citric acid cycle because we start with oxaloacetate joining with acetyl-CoA, and after one turn, we regenerate this oxaloacetate, which can then enter again. Okay, so that's why it's called a cycle. So in the citric acid cycle itself, what do we generate? We generate, remember this is going to happen twice because we get 2 acetyl-CoA molecules. We will get 4 carbon dioxide, 2 per turn. We would get 1, 2, 3, 6 NADHs, 3 per turn, okay? 6 NADHs, 2 FADH2s, 1 per turn, and we would get 2 ATP, right? 1 per turn. So out of all of this in the citric acid cycle, we only get 2 ATP if we have both pyruvic acids entering into the mitochondria. Only 2 ATP. So far, we have gotten 2 ATP from glycolysis and 2 ATP from the citric acid cycle, 4 ATP total. That's not a whole lot, right? So we have to go through all of this work for only 4 ATP, but what have we generated a whole lot of? NADHs, right? We got 2 NADHs from glycolysis, and we got 2 NADHs from this step, 6 NADHs from 2 turns of the citric acid cycle, all right? So that means we have 6, 7, 8, 9, 10, right? 10 NADHs that we have synthesized from glycolysis and the citric acid cycle. Wow, that's a lot of NADHs. We've also generated a few of these FADH2s. So the point of the citric acid cycle is not to make ATP directly because we've only made 2 of those. The point of the citric acid cycle is to generate a lot of these high energy electron carriers because that's what we're getting the most of, right? The point of the citric acid cycle is to generate a lot of these high energy electron carriers so that these can then enter into the citric acid cycle, and that's how we get the bulk of our ATP, is through the electron transport chain. So the point of the citric acid cycle, which takes place in the mitochondria, the matrix of the mitochondria, is to generate a lot of these high energy electron carriers that can then enter into the electron transport chain, which is embedded into the inner membrane of the mitochondria, and that is where we get the bulk of ATP synthesis, okay? So what have we made so far? In glycolysis, we made a net 2 ATP, 2 NADHs, and 2 pyruvic acids, okay? Those pyruvic acids then enter into the mitochondria and get converted into acetyl-CoA. Each pyruvic acid gets converted into an acetyl-CoA molecule. And in this step, we will generate 2 NADHs, 2 carbon dioxides, 2 acetyl-CoAs, right? Each one of these acetyl-CoAs can then enter into the citric acid cycle, and we would get 2 turns of the citric acid cycle. Through the citric acid cycle, we would generate 1, 2, 3, then the second time 4, 5, 6 NADHs, 6 NADHs, 1, 2 FADH2s, 2 ATPs, and 4 carbon dioxides. Okay? And remember the carbon dioxides, this is just a way to completely oxidize that glucose, and the carbon dioxide will eventually get exhaled as a waste product. Okay? So we haven't made a whole lot of ATP, but we have made a lot of NADH molecules and FADH2s. These high energy electron carriers that can then go to the electron transport chain where we can make a lot of ATP. That is the point of the citric acid cycle.